Hello friends, welcome back to my channel Medicine Hub 101. Today we'll talk about insulin synthesis, secretions or regulations, or you can say the regulation of insulin synthesis and secretions. Okay, before starting the video, let me tell you I have already uploaded one video where I have explained in details that how the insulin that will be formed, now how the insulin that will be synthesized and how it will be secreted. Okay, so I'll just do the rapid review of the first video, then I'll go in details about this part. Okay. So whenever I said that after taking the diet, so when you are taking the food, the glucose that will be absorbed, right? The glucose will get the glucose molecules that will be absorbed where within the circulations. First we'll get from here, it will come within the circulations, and from the circulations we'll get, okay, we'll go where we'll get within the circulations, and you got the glucose within the circulation, and after that what other thing we'll see. See, whenever we are getting the glucose over here, this glucose that will be also transported to the what? Pancreatic beta cells. And I'm just drawing a pancreatic beta cells over here. Okay. I'm just drawing the pancreatic beta cells over here. As I said earlier in the first video, that how the insulin that will be formed. Okay. Now see, I mentioned you over here that within the pancreatic beta cells there is a presence of the GLAD2 transporter. We can say GLAD2 transporter. So if you can say over here that we will get the GLAD2, this is the GLAD2. Now this GLAD2, what they will do, whenever this glucose that will come over here, and they will just take the glucose and they will flip in. That means they will take the glucose and they will just do the flip in. As a result, we will see the glucose that will be taken within the pancreatic beta cell. So after that, we will get the glucose over here. And as we know that whenever the glucose that will be taken with the cells, there is chances for the glucose to go back to the outside of the cells. That's why one type of the enzyme will come over here. You know, the glucokinase enzyme. Sorry, the glucokinase enzyme that will do what? Glucokinase that will just do the additions of on phosphate along with this glucose molecules. Now it is getting the charged molecules, or you can say the polar molecule. Okay, and the next process you know from the basic biochemistry that we will get the productions of ATP by the TCA cycles, by the glycolysis process, where we will get the conversions of ADP plus phosphate into ATP. Is the normal thing we know, right, from the basic knowledge. Now see, there is a presence of one type of the potassium channel, leaky potassium channel. There is a presence of one type of leaky potassium channel who are within the pancreatic beta cells. Now under normal circumstances, if you see there is no glucose, it's normally okay. If we see there is no glucose, no energy, at that time we will see the the potassium, they will be freely permeable from the inside of the cells to the outside of the cells. So we will see the freely potassium outcoming, or efflux of the potassium. We will get the efflux of the potassium, and in this status we will see there is a presence of on site or on pocket within the channel that is known as site for the ADP. So try to understand whenever the glucose is not within the cell at that time, definitely the ATP that will be less. So whenever the ATP is less, definitely the ADP is more. That means whatever the amount of the ATP was within the cells, all of these ATP molecules already got broken down. Now as a result we will get the ADP that will be more, the ATP that will be less. Sorry. Now, here in this cases, what are the thing we'll get, that there is a site here the ADP can bind. So whenever the ADP there that can bind over here. So after binding the ADP, adenosine diphosphate, we'll see the potassium that will be freely permeable from inside of the cells to the outside of the cell. Now we are getting the productions of the ATP molecules. Okay, we are getting the production of ATP molecules. Now, whenever you are getting the productions of the ATP molecules, what type of the changes we will see over here? Try to understand from these portions. These ATP molecules that is produced whenever we are getting the glucose, they will come over here and they will just do the blockage of the ATP molecule. They will just do the blockage of the ATP molecules. sorry, blockage of the potassium channel by the ATP molecules. This ATP that will bind along with this potassium channel, and this ATP that will block the potassium channel. So whenever the potassium channel that will be blocked, as a result we will see the potassium that will be not able to go outside of the cell. So the potassium that will be repelled back. As a result, within the cell, or you can say intercellular potassium concentration that will be increased. So whenever it will be increased, for example, whenever it was going out, at that time we got the resting membrane potentials or the hyperpolarizations for the leaking out of the potassium. Now whenever we are getting the ATP is binding along with the potassium channel and it is not letting the potassiums to go outside of the cell. Sorry. As a result, we will see the resting membrane potential that will be neutralized. Whenever it will be neutralized, for example, it can reach up to the threshold potentials, for example, okay. So whenever it will reach up to the threshold potential, at that time we know the voltage sensitive calcium channel that will be activated, right? The voltage sensitive calcium channel that will be activated. And whenever the voltage sensitive calcium channel that will be activated, we will see the huge influx of the what? Calcium. So we'll get the calcium within the cell. So whenever we'll get the calcium within the cell, this calcium what they will do, as I said to you before already, that we'll get within the beta cells, within the beta cells we'll get some vesicles, within the vesicles we'll get the insulins that will be synthesized and stored. Now what this calcium they will do, this calcium they will take the vesicles, they will take the vesicles and they will fuse along with this membrane. So whenever you will see this, you will fuse this one with this membrane. What type of thing we will see? After that we will see by the exocytosis process, we will see the releasing of the insulin. Along with the insulin we will get what? C-peptide and some amylin, right? Some amylin. So this is a normal process how the insulin is getting stored and how the how the insulin is getting when you are getting the glucose within the pancreatic beta cells. Sorry guys. Now we'll talk about that there are certain kinds of pharmacological agents that can stimulate, that can inhibit the ATP sensitive potassium channel. So if the ATP sensitive potassium channel that can be blocked, if we can block by the pharmacological agents, if we can block the ATP sensitive potassium channels, then this potassium channel that will be blocked. So whenever it will be blocked, definitely the within the cells will see the the resting membrane potential that reach to threshold potentials, voltage sensitive calcium channel will be opened, we'll get the calcium influx. So as a result, we will get the productions of the insulin. Now we can use certain kinds of the drugs. Okay, so you can write it over here that ATP sensitive potassium channel where within the pancreatic beta cells, blockers or inhibitors. So what type of the drugs we will get over here? At least two groups of the drugs we are supposed to know. One is known as sulfonylurea. Now see, this sulfonylurea, what they will do, they will go over here and they will just block the ATP sensitive potassium channel. As a result, we will get the potassium that will be within the cells, or you can say the intercellular potassium concentration that will be increased. Another group of the drugs that is known as meglitinide. So the meglitinide and the sulfonylurea, they are the meglinitide, right? Meglinitide. Sorry guys. So sulfonylurea and the meglinitide and sulfonylurea, they are the group of the drugs that will inhibit the potassium, ATP sensitive potassium channel. So whenever this channel will be blocked, definitely we will get the outcoming of the insulin. Now another group of the drugs, there is another group of the drugs that will stimulate the what? That will stimulate the potassium. For example, if we see any kind of the drugs that will stimulate the potassium and that will help to efflux of the potassium. See, whenever see the potassium efflux that will be increased, definitely the insulin production that will be less. What I am saying, try to understand. If we develop any kind of the drugs, okay, if we develop any kind of the drugs that is stimulating the this ATP sensitive potassium channel, or you can say simply the potassium channel, stimulate the potassium channel. Whenever the potassium channel that will be stimulated, we will see the potassium concentrations, the potassium that will leak out outside of the cell, right? Leak out the cell, leak out of the cell. So whenever the potassium that is coming out of the cell, as a result, we will see the cells that will be what? Hyperpolarization. Within the cell, we will get hyperpolarization. So whenever we will get the polarization. So whenever we are getting the hyperpolarization, try to understand definitely there will be no activations of the voltage sensitive calcium channels, so there will be no productions of the insulin. Okay. What are the drugs are those? The name of the drugs that is known as dioxide. Dioxide. So dioxide, what it will do? It will just what its main functions basically we use the drug in terms of the antihypertensive drugs, right? Why? Try to understand from here. Basically, this drug that is used in terms of the antihypertensive drugs, dioxide. Now, what it will do? It will also activate the potassium channel that is present within the blood vessels, right? Within the blood vessels or within the smooth muscles, right? For example, if I say over here is the blood vessels will be over here, and I'm just considering this is the smooth muscles. Okay, let's draw it in the better way. Okay, for example, is the smooth muscles. What it will do, this dioxide that will go over here, that will go along with the water within the blood vessels within the potassium channel. So whenever we'll see the presence of the potassium channel over here, and they will activate this potassium channel. So whenever the potassium that will leak out, as a result, we will see within the smooth muscular cells, we will get what the hyperpolarizations. So whenever we will get the hyperpolarizations, at that time we will see the voltage sensitive calcium channel that will be not activated. So whenever the voltage sensitive calcium channel that will be not activated, that will be blocked. So whenever the voltage sensitive calcium channel that will be blocked, so definitely we will not get the influx of the calcium. So influx of the calcium that will be inhibited. So whenever the influx of the calcium, influx of the calcium that will be inhibited, definitely the smooth muscles they will be not able to contract. So whenever there will be not contractions, we will get rather what the dilations. So the vasoconstriction that will be inhibited by this drug, rather will get what the vasodilations, right? That's why. So whenever we get the vasodilations, definitely the blood pressure that will fall down, right? So we can see in another way that this dioxide that will act as also as vasodilator. Vasodilator. So what about this one? This dioxide that will be used if, for example, if someone is having a tumor, for example, the tumor production in the insulin production tumors, for example, we can see in terms of the insulinoma. So if we see any individual who is developed, any individual who has developed the insulinoma, at that time we will see the productions of the too much insulin. So whenever there will be productions of the too much insulin within the blood, definitely we will see the development of the hypoglycemia. Now to treat these things, we have to use the dioxide. Now whenever the dioxide that will go over here, and that will just that will go and that will activate the potassium channel. We will see more amount of the potassium efflux. So whenever more amount of the potassium efflux, we will see over here. Try to understand. So definitely we will not get the what? We will not get the depolarizations over here. So we will get rather we will get the hyperpolarizations. So ultimately we will get the less insulin that will be secreted, less C-peptide, less C-amylin, or you can say amylin, sorry. So whatever the drugs, that means we can see in terms of the dioxide, we can use in terms of the antihypertensive cases, and we can also use in terms of the insulinoma. We can get whenever we will get the tumor within the pancreatic beta cells. Okay. Sorry guys. So this is the drugs we can use over here. Okay. Now whatever another things we have to know over here, that during eating whatever the other things, for example, as I have said you guys that this glucose that will activate the pancreatic beta cells, there are also other factors that can also activate the pancreatic beta cells in terms of the different kinds of the hormones. So let's talk about those things. Now whenever you are eating food, whenever you are taking food, by taking too much or a lot of food, what are the thing will see over here right after that? See, whenever you are taking food, try to understand from here, from the GIT from the GIT from the we can say, or we can say that from the GIT epithelium, from the epithelium of the GIT wall. Okay, we can write from GIT wall, within the GIT wall, from where? From the epithelium. Whenever you are taking food, during taking food or after taking meal, whatever the thing we'll see over here. So when you are taking the food, at that time, for example, you are taking the glucose molecules, or are using the proteins or the fatty acids, fats, whatever it is. Now whenever we will take the food, at that time you will see from the GIT wall, from here the epithelial cells, they will secrete a group of peptide hormones. Okay, so they will secrete a group of peptide hormones from these cells. So group of peptide hormones. This group of the peptide hormones, they are known as what? Incretins. They are known as incretins. What is the function of the incretins? Sorry. The functions of the incretins, they will activate the beta cells to release additional amount of the insulin. So the first thing you can write it over here. The release of insulin, or releasing of, we can say releasing of insulin. Why it will do so? See, whenever the insulin that will be released, okay, by this mechanism. So whenever insulin that will be released, the insulin, whatever the for nutrients that is present in the circulations, just to make the circulations with normal nutrients level, the insulin that will deposit the different kinds of the nutrients were in the different kinds of the cells, as we know from the basic knowledge. So this incretins, they will release the insulin, they will help to release the insulin. Along with that, as we know that whenever the insulin is supposed to be secreted, at that time the glucagon, the glucagon hormone, that is supposed to be suppressed. So what it will do, it will reduce the production of the glucagon. Okay. Another thing, what it will do, the incretins that will reduce the motility of the stomach. The incretins that will reduce the motility of the stomach. See why it will do so? Because if we see, the motility of the stomach is not regulated properly. As a result, we can see the overflow of the glucose within the small intestine. So the digestion that will be not done properly. Now just to do the digestions properly, the motility of the stomach, that should be regulated properly. Okay, so we can say rather that that the motility of stomach, or regulation, or we can say in another, it will be inhibited. Inhibited means in this way, it will just inhibit the overflow. It will just inhibit the overflow from the stomach to the where? Within the small intestine. As a result, we will see the digestions that will be done properly from these portions to the what's within the circulation will get. And another thing, whenever we are taking the enough amount of the food, definitely after that, if anybody tells us that we want to take more food, or if someone, for example, have taken a lot of food, we have taken a lot of food, and when someone is asking us that do you want guys more food, then what we are supposed to say at the time? No, it's enough for me. Why? Because whenever the incretins is getting secreted, at that time it will go to the hypothalamus and it will activate the satiety center of the hypothalamus, right? So it will activate the satiety. So whenever the satiety center that will be activated, after that we will not feel too good. We will not feel to eat any amount of the more food. So we'll say yes, it's enough for us. We don't want to take any more food. Okay. So what are the main functions? Releasing of the insulin. In fact, we can say insulin is getting secreted by the what? Pancreatic beta cells after taking the glucose. So it is just in amplifying for the releasing of the insulin. We can say also the amplifying of the releasing of the insulins. Okay. Then it will inhibit the secretion of the glucagon. It will reduce the motility of the stomach. As a result, we will see the digestions that will be done properly. Along with that, it will have the satiety center of the hypothalamus. So these are the functions in terms of the incretins. Now, what are the incretins? As I said, the group of the peptide hormones. What are they? So what will be the incretins? Incretins first will get, that is known as glucagon like peptide on. Don't get confused with the glucagon. I'm writing it over here, then I'm explaining. Glucagon like peptide 1. Don't get confused. This is known as also GLP-1. GLP-1, glucagon-like peptide 1. Okay. Don't get confused with the glucagon. The glucagon that will be secreted from the pancreatic alpha cells, and this glucagon-like peptide 1 that is getting secreted from the GITol, from the GITol within the epithelial cells. Okay. Now, what are the other hormones? Okay, let's use the different color, I think, for it. Okay, it's not working. Another hormone that is known as gastric inhibitory hormone. Gastric, from the term, what did I say? The incretins, they will reduce the, they will inhibit the GI motility. That will just reduces the GI motility. We can say the stomach motility. As a result, the proper digestions will be found over here. Now, that is another hormone under the incretins that is known as gastric inhibitory peptide. Inhibitory peptide. It is also known as glucose dependent. Okay, insulinotropic glucose uptake. Okay, we can write over here. Another name for it, that what? Glucose dependent insulinotropic. Dependent insulinotropic peptide. Another name for it. So if you want to write, you can write it over here. That glucose dependent, I am writing over here, glucose dependent insulinotropic peptide hormone. Glucose dependent insulinotropic peptide hormone. Okay, I just did repeat 2 to 3 times. You are supposed to listen and you can write it over here. Another hormone you can get in terms of, that cholecystokinin, this type of hormones will get. But the most important 2 ingredients you are supposed to know, that the glucagon-like peptide on, another is what? Gastric inhibitory peptide, or we can say also that glucose-dependent or insulinotropic peptide. Okay. Now we talk about certain types of the enzymes and the drugs related to the incretin. Sorry. Now see the glucagon-like peptide on and the gastric inhibitory peptide in terms of the incretins, how do they work? Okay, we'll talk about all these things. Before that, I have to erase some part from here, guys. Okay, from here should I erase this thing? Okay, I'm erasing this part. Now, what do you know in terms of the incretins? As I said, that incretins, for example, in terms of the GLP-1, the glucagon-like peptide 1, if you can make certain types of the drugs, since if we can get certain types of the drugs, similarity for the GLP-1, okay, if we can make some drugs like the GLP-1, but whenever we use the drug, what type of problem we will see, if we just make GLP-1 like drug, what type of problem we have to face? So whenever we make this type of the drugs, whenever this drug, they will come within the circulation, within the circulations there is a presence of one type of the enzyme that is known as, okay, let's use the different color because it's important. So as I said, if we can develop the GLP-1 like drugs, that means glucagon like peptide 1, if we can develop drugs, then for example, whatever problem you have to face over here, there is a presence of an enzyme within the circulation that is known as dipeptidyl peptidase 4, or you can say DPP4. So whenever we will see the development of the drugs like the GLP-1 or glucagon like peptide 1, whatever the problem you face within the circulation, there is a presence of one type of the enzyme that is known as dipeptidyl peptidase 4 that will destroy the these drugs, the GLP-1 like drugs. So as a result, the scientists that develop the GLP-1 like the drugs, or you can say the synthetic GLP-1, for example, in these cases we will get the drugs that will be developed that will be slightly different from the original GLP-1. The drugs will develop, the synthetic GLP-1 will develop, that should be slightly different from the original or the natural GLP-1. So whenever we will see, there will be slight difference within the, slight difference within the drugs. As a result, this enzyme, they will be not able to destroy the drugs. Okay, so what are the drugs, we will write it over here. Before that, okay, let's continue. Okay. So what I was saying you, yes. So what I was saying you that if you can develop the certain kinds of drugs, okay, let me write it. Then I'm explaining these things. Okay. Now see, if we can develop certain kinds of the drugs like the GLP-1, the glucagon-like peptide on, but these drugs would have some dissimilarities. Like we can say these drugs would have the similarities changes. That means we can say this drug is not supposed to be structurally and functionally same as the GLP-1, because if it is the same, it is supposed to be same, but there should be slight difference. There should be slight difference in terms of the drugs GLP-1 or the normal, or we can say the original GLP-1. So in the drugs exenatide, we can use the drug exenatide. That's what whenever it will be within the circulations, the DPP4, or you can say the dipeptidyl for peptidase 4, they will be not able to destroy these things. So these things, they will go where within the pancreatic beta cells, and they will release more amount of the insulin. They will act as a what? They will act as amplifier. Now another kind of the drugs, for example, another kind of the drugs, if you can develop any kind of the drugs that will inhibit these enzymes, that will inhibit the enzyme, that is what? That is what? Let me tell you, let me repeat it once again, that if you can develop any kind of the drugs, okay, that will inhibit this enzyme, that means dipeptide phosphatase, oh sorry, dipeptidyl peptidase 4. So let's write it over here. The drugs inhibit the DPP4, that means dipeptidyl peptidase 4. If you can develop certain kinds of the drugs, dipeptidyl peptidase 4, whenever you get the inhibition of dipeptidyl peptidase 4, what type of the drugs we can use over here in terms of the sitagliptin, okay? So we can use sitagliptin. Sitagliptin. Now whenever we are using the sitagliptin, what they will do, they will inhibit this enzyme. So whenever the DPP4 that will be inhibited, as a result we will see within the circulations, we will see the more amount of the incretins level, that will be elevated. So whenever we get more amount of the incretins, as a result, as I said you over here, the incretins, they will just do what? They will increase the secretions of the additional insulin. More amount of the insulin that will be secreted. So insulin secretions that will be increased, or that will be elevated over here. Okay, get in this way. Now, as I said, we can use the same type of the drugs, just like the incretins in terms of the GLP-1, glucagon like peptide one. We can develop certain types of the drugs, exenatide. That these drugs would have the some structurally dissimilarities along with the what GLP-1. It is not supposed to be exactly we can say GLP-1. As a result, this DPP4 that will be not able to destroy these things. Like this, we can increase the secretion of the insulin. Another kind of the drugs we can use over here, the sitagliptin. Whenever we are using the sitagliptin, that is inhibiting the DPP4, or we can say the dipeptidyl dipeptidyl peptidase 4. So as a result, the incretins level that will be increased. So the more amount of the insulin that will be produced over here is the thing. Okay. Now we'll talk about certain kinds of the hormones. How do they activate the different kinds of the secretion of the insulin over here? Okay, let's talk about those things. For that thing, I have to erase some part from here. Now we'll talk about all these things. Now, for example, here is the pancreatic beta cells. Here is the pancreatic beta cells. Now, the glucagon hormone that can also activate the pancreatic beta cells to release more amount of the insulin. How? Let's see over here. So, if we get the glucagon hormone over here, this glucagon, what they are going to do over here within the within the pancreatic beta cells, there is a receptor for the glucagon. There is a 7 pathway receptors. So whenever this glucagon will go over here, and that will get bound along with the glucagon receptors, and as we know, this is the G stimulatory. So whenever this G stimulatory receptor that will be activated, that will give signal within the adenylyl cyclase enzyme, and whenever this enzyme will be activated, that will convert the ATP into what? Cyclic AMP. So whenever we will get within the pancreatic beta cells cyclic AMP, that will be increased. This increased cyclic AMP that can facilitate, or that can help to productions for the, whenever we will get within the pancreatic beta cells the cyclic AMP, that will be increased, and that will secrete more amount of the insulin. whatever the cause of mechanism. So we can say over here. In the same way, we will also see the incretins, what they will do over here. Try to understand. In the same way, the incretins, you know, the incretins, they are also having, the receptors. The incretins, they are also having the receptors. Serpentine receptors over here also, and they are also G-stimulatory. In the same way, we will get, they will also activate the what? Adrenaline cyclins. In the same thing, we will get, and we will get also the conversion of more amount of the ATP into cyclic AMP, and we will also get the more productions of the cyclic AMP. So more productions of the cyclic AMP, that leads to productions of insulin hormones. Okay, these are things. So we can say the glucagon hormone that can activate these things. Along with that, the incretins, how they are also having the G-stimulatory receptors within the beta cells, and they are also increasing, or you can say the conversions, or they are also doing the conversions of the ATP into cyclic AMP. We will get the more productions of the cyclic AMP, and the more produced cyclic AMP, that leads to productions for the more amount of the insulin hormone. Okay. Now we will talk about the other kinds of the hormones. Now we will talk about, for example, any individual who is having, who is like the acromegaly. The acromegaly person, whatever the problem we see in terms of the acromegaly. See, in terms of the acromegaly, whatever thing we see, there is a production of the more amount of the growth hormone. So whenever we see the more productions of the growth hormone, what they will do, this growth hormone, they will increase the gluconeogenesis process. Gluconeogenesis process. So this growth hormone that is increasing the gluconeogenesis process, as a result within the blood will get the glucose level that will be elevated, right? The glucose that will be elevated, will get the elevations of the glucose within the blood. Along with that, this growth hormone, what they will do, they will reduce the peripheral uptake of glucose. They will reduce the peripheral uptake of glucose. So they will reduce the uptake of glucose. As a result, within the blood will get the glucose level that will be elevated, and this thing is known as what? Hyperglycemia. You will get hyperglycemia. In the same way, we will also get for the cortisol. If we get, or in the Cushing syndrome, so we will get the more productions of the cortisol. The cortisols, they will also do the same mechanisms. They will also do the gluconeogenesis, and they will also reduce the peripheral uptake of glucose. As a result, that will also leads to the hyperglycemia. Okay. Now, what about these things over here? Yes, another thing, important thing that I'm supposed to say this thing. See, if we see any patient who is having the glucagonoma. If we get the tumor within the glucagon producing cells, that is known as glucagonoma. So whenever we get the development of the glucagonoma, at that time we will see this glucagon hormone that will give, that will be overstimulated, and whenever this glucagon we will get, because see, whenever we are getting the glucagonoma, at that time we will get the more productions of the glucagon hormone. So this more produced glucagon hormone, that will overstimulate the glucagon receptors. Whenever it will be overstimulated, we will get, as a result, we will get the more conversions of the ATP into cyclic AMP. So whenever we will get the more productions of the cyclic AMP, that will leads to more production of the insulin. But if we see this, overstimulations for longer durations, at a time, the pancreatic beta cells, that will get exhausted. So whenever it will be exhausted, after that, at a time we will see this pancreatic beta cells, that will be dysfunctional. So over here we can write, there will be the overstimulation. Whenever we get the overstimulation, try to understand from here, the beta cells that will be exhausted. So whenever it will be exhausted, try to understand from here, whenever it will be exhausted, at that time it will ultimately get the dysfunctional. So whenever it will be dysfunctional, definitely we will get the development of the hyperglycemia. We will get hyperglycemia. Now we'll talk about in terms of the, there are certain kinds of in terms of the central nervous system regulations, autonomic nervous system regulation. So how we will see the autonomic nervous system regulation that will regulate these things. Okay, how it will regulate. Now for that thing, if we write it over here, automatic nervous system. Automatic nervous system we can divide into two ways, right? At first we can write in this way that the parasympathetic nervous system and sympathetic nervous system. So see, whenever the parasympathetic nervous system, it will be activated. See, under resting conditions, right? This parasympathetic nervous system that will be activated. And what? The sympathetic nervous system under the stressful conditions. So at first I'll talk about the parasympathetic nervous system, then I'll go back to the sympathetic nervous system. Okay. Now see, what are the things? So whenever you are saying that under resting conditions, for example, you have taken the food. Whenever you are taking the food, at that time the vagus nerve that will be activated. So from the parasympathetic here, from the parasympathetic nervous system, we'll get the vagal outflow. Vagal outflow. Whenever we will get the vagal outflow, from the nerve endings, we'll get the releasing of acetylcholine. We'll get the releasing of acetylcholine. So whenever we get the releasing of the acetylcholine over here, it will activate these cells, and as a result we will get the productions of the more amount of the cyclic AMP. So whenever we get the productions of the more amount of the cyclic AMP, that will leads to more productions of the more amount of the insulin. I am repeating once again. As far as the central nervous systems, or you can say the autonomic nervous system, sorry, you have to say in terms of autonomic nervous system regulations, how they can regulate the insulin productions. Whenever say under resting conditions, whenever you are taking the food, at the time the parasympathetic nervous system that will be activated, right? Now that will, from here will get the vagal outflow, and as we know that during eating conditions or during the feeding conditions, we will see the vagus nerve that will be activated. So we'll get the vagal outflow that will release the acetylcholine, and this acetylcholine neurotransmitter that can activate the pancreatic beta cells to produce more amount of the cyclic AMP. Whenever we are getting the more amount of the cyclic AMP, we will get definitely more productions of the more amount of insulin hormone. Now in the same way, if we say in terms of the sympathetic nervous system, in terms of the sympathetic nervous system, what are the thing we'll see over here? I mean, see, in terms of the sympathetic nervous system under stressful conditions, for example, someone is having the, someone is suffering from fever, or someone is someone is ready for going to the surgery, or if you get any kind of the physical trauma, at that time we will see the sympathetic nervous system that will be activated. So we can say over here under stressful conditions, right? When it will be activated under stressful, under stress, will see sympathetic nervous system that will be activated. And whenever see sympathetic nervous system that will be activated, at that time whatever the thing we'll see over here. Okay. See, whenever the sympathetic nervous system that will be activated, that will release what? That will release the norepinephrine. And as we know, within the pancreatic beta cells, there is the presence of alpha-2 adrenergic receptors for it. We get what? Alpha-2 adrenergic receptors. So these alpha-2 adrenergic receptors that will be activated whenever the norepinephrine that will come over here. And as we know, this is the G inhibitory. This is the G inhibitory, right? This is the G inhibitory. And whenever we are coming to know the G is the G inhibitory, what it will do? It will, as you know, it will just inhibit the adenylyl cyclase. So whenever the adenylyl cyclase that will be inhibited, definitely we will also get the inhibitions to produce from the ATP to cyclic AMP. So this process that will be inhibited. So whenever this process that will be inhibited over here, conditions, the sympathetic nervous system that will be activated. Whenever it will be activated, it will release the norepinephrine. And within the pancreas, there is the presence of alpha-2 adrenergic receptors. So this norepinephrine that will come over and that will get alpha-2 adrenergic receptors. And as we know, this is the G inhibitory. So we'll see over here that will get the inhibitions of the adenylyl cyclase enzyme. So whenever the adenylyl cyclase that will be inhibited, so definitely we will get also the inhibition of in conversion of cyclic AMP, so ATP to cyclic AMP. So cyclic AMP is getting reduced. So definitely we can say the insulin production that will also reduce. This is a normal thing. Okay. Now another thing, if we say from the adrenal medulla, as we know, the adrenal medulla. Okay, let's so over here. From the adrenal medulla, okay, let's use the different one. Then I think it will be more clear for you. Yes. So from the adrenal medulla, what do you know? The releasing of epinephrine. Try to understand. Basically, the epinephrine that will be released from, in most of the cases we see the epinephrine that is getting released from the adrenal medulla, but norepinephrine from the sympathetic nervous outflow, from the sympathetic nervous system outflow will get the releasing of the norepinephrine. But whenever you are getting the releasing of the epinephrine from the adrenal medulla, whatever thing they were going to do, okay, in these cases we will get there is the presence of what? Beta 2 adrenergic receptors, beta 2 adrenergic receptors that is present over in the pancreatic beta cells. So whenever this epinephrine that will go over here, that will get bound, and is the G stimulatory, is the G stimulatory. So it will go, it will just, it will activate the adenylyl cyclase, it will activate it, and it will convert the what? ATP into cyclic AMP, right? ATP into cyclic AMP. So whenever it will be converted from ATP to cyclic AMP, we will get the more productions of the insulin also over here. So this is the thing we can get over here. Okay. Now, as we know that on the beta, on pancreatic beta cells, the concentrations of alpha 2 adrenergic receptors more than the beta 2 adrenergic receptors. What did I say you? That on pancreatic beta cells, if I write it over here, that on pancreatic beta cells, the concentrations of alpha 2 adrenergic receptor is more than the beta 2 adrenergic receptors. So definitely we will get the more effectiveness for alpha 2 adrenergic receptor, that means in terms of the sympathetic outflow, sympathetic nervous outflow. Okay, that's the thing. Now there are also certain types of the hormone that is known as somatostatin. The somatostatin is the peptide, hormone inhibitors. We can say the somatostatin that will inhibit all types of the hormones. It can inhibit the GLP-1, like, we can say the glucagon like one peptide hormone over here, we can say, or we can say the 6-CCK, the cholecystokinin, hormones, all these hormones, or you can say over here also the gastric inhibitory hormones that can inhibit also all these hormones. By inhibiting all these hormones, it will try to contain these, maintain the situations. But that's the next thing I'll talk about, the whenever I'll talk in details about the management in details about this whole thing. I'll just mention all these things over there. So this was all about the regulation in terms of the synthesis, secretions, insulin synthesis and secretion. Next part we'll talk in details about the more pharmacological agents. And if you get the, for example, if you get the diabetic ketoacidosis or diabetic coma, at that time, how can we just manage these patients? What we are supposed to do? At first, we'll give the fluids, then we'll just inject the insulin. After that, we'll just give what? We'll just do the potassiums from outside. So how can we do this? All these things in terms of the management in the severe cases. We'll talk about all these things in the subsequent parts. Okay, if you think this video is helpful for you, please do like and subscribe my channel. And see you in the next video very soon with the more complex cases. Thanks for watching.